Okay, how's it going, class? So today we're going to talk about a harder subject. We're going to talk about how does DNA, how you start with DNA and create a protein. Well, the first thing we have to ask is, we know what DNA is. We know that's the blueprints for all of us. But the question we ask is, what is a protein? What's the point of proteins? Well, what a protein does is it pretty much tells everything about you. It tells what color you're going to be, what color your eyes are going to be, your hair is going to be, how big you're going to be, how big your muscles are going to be, how big different organs, how they're going to function. Proteins pretty much tell your body how to function. There's many different types of proteins, and from DNA we can get these different proteins that create the big us. Now, an essential thing to understand is that proteins are simply made up of something called an amino acid. Amino acids make up proteins. And how they do this is, the way we're going to show you is we have these little balls right here, and they're all just floating around by themselves. Each one of these represents an amino acid. And what a protein isn't an amino acid by itself, but when you link these things up, I'll put a little bond between all of them so they're all friends in a nice line. When we link these things up, it becomes a protein. So the question is, how do we get these amino acids into a straight line and in the correct order for what the protein has to do? Because let's say we need to make a protein that carries out replacing skin cells. That helps cells to repair, like if you got a cut. We can't just hope that all these amino acids will kind of group in the right way, because what if there's a mistake and we get a mutation or something even worse? Well, your body has a pretty good way of using those blueprints of the DNA and eventually changing it into amino acid, and then taking these amino acids, and then turning it into a nice usable protein. Let's talk about how this works. So what will first happen is you're going to have your DNA, and I'm going to show you this, and I'm going to show you the pictures. You're going to have your DNA, and in our DNA we're going to see a genetic code. And remember, this is taking place in the nucleus. So all this right now is taking place in the nucleus. And in the DNA, your body will say, hey, we need some actions to make some new skin so we can repair the damage done when you got cut. So it finds the part of the DNA that codes for your skin, that codes for how to repair it. So let's say it's looking along the DNA and it comes across this sequence and it realizes that this is indeed the correct sequence in order to create a protein that can help fix your skin. So what it'll do is something will come in, because remember, the DNA can't leave the nucleus, and proteins, if you remember, proteins are made in the ribosomes. And there's a problem. Ribosomes aren't in the nucleus, they're actually in the cytoplasm. So say here's the edge of the nucleus, here's the cytoplasm. How do we get that information here? They can't shout it, they don't have ears or mouths to say it. Instead, what comes in and sits right along here is called mRNA. And this M stands for messenger, because it can come in, take the message, and then bring it out to the ribosomes. Well, how does mRNA work? Well, what mRNA does is it comes, finds that sequence on the DNA, and then it starts matching its own bases against that. And if you remember, in DNA, if we were looking at TACCCG or GGAAT, the matching sequence that would be what? ATGGGCCTTA. But mRNA uses something different. Instead of using T's, it uses something else. It uses U's, and that U is called a uracil. And uracil instead replaces the thymine. So when we look at this, the T and A stay the same, so it'll still use A, C, and G, but it won't use the T, it uses a U for uracil. So when we look at this, the T, it'll still translate over to an A, but this change, instead of being a T, it becomes a U. And then this one becomes a G, just like normal. G, G, C, C. Alright, this A, remember, you can't use T's because this is RNA, and RNA does not use T's. Instead, it uses U, U, and then this one, remember, T can still say the same as going with here. So now this mRNA has a strand that says AUGGCCUUA. It can then take this, because remember, the mRNA can leave, so it leaves the the nucleus, leaves the DNA, and the DNA will find its pair, because remember it was taken apart so it could copy this. It goes back together and forms back into a double helix, completely unharmed. The mRNA comes down here and sits in the ribosome. Now with this new sequence, A, U, G, 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 C, C, U, U, A, with this new sequence, what it can do now is it can sit inside the ribosome, and it kind of goes along the ribosome in this direction. So it kind of filters through. It's like the ribosome's pulling a rope of it through. And as it pulls it through, the ribosome will do it by threes. It'll alert a letter, it'll say, hey, I have an AUG. And that means nothing to us, except for the fact that there's something called a tRNA floating around in the cytoplasm. So you have a tRNA. Now this tRNA is called a transfer RNA. And with this floating around the cytoplasm, it'll recognize an AUG. Because on the tRNA, it'll have these little hooks on it, and you can probably guess what's on it. It's probably its matching pair. And matching with that AUG all of a sudden, it might have a UAC. Because remember, that's going to match up. Because remember, it's an RNA, so it doesn't use T. So this UAC comes over, and it finds that spot, and carrying on top of the tRNA is an amino acid. So I'll put AA for amino acid. And it goes into the spot, locks it onto there, and drops off its amino acid. Then that moves through because, and the tRNA will leave because he dropped off his amino acid. Then the ribosome will ask for a GGC. Well, another tRNA comes right after, and he has the matching sequence for that. Now again, it's not going to be GGC again. Remember, it has to be the opposite. It has to be so it can lock into place. And so it's going to be having a CCG. And he also has a specific amino acid, and he drops that one off. And so this code keeps going, and it keeps locking all these amino acids together, eventually creating a protein. Now let's look at this in pictures. And you can follow along in your book. Again, this is like the homework that we had last night on page 116, 117. And you'll see it here in your book. Let's bring that up. Now right here in our book we can see that, and if you need to pause it so you can open your book. We can see that first step I was talking about. The mRNA enters the cytoplasm, so it needs a type of protein. Well, the DNA, like I said, unzips. It doesn't unzip the whole thing. It's not replicating. It just opens it so this mRNA can fit in there. And we have a TGT-GAA. So the mRNA comes in here and says, hey, I need that code. So it'll go ACA, CUU. And you can see how it kind of fits into place. You couldn't have this C fit over on the A because the shape wouldn't fit. And the DNA makes sure it doesn't screw up. Because remember, only A can go to U, G can go to C, and the original T can go to A. Remember, because this U shows up because we can't use T's in the RNA, but we still have these T's in the DNA that will code for A's. So this messenger RNA is now allowed to leave the nucleus, and it finds a ribosome. This blue thing right here is called the ribosome. Now as it goes through, remember it's like feeding through, so it shows the ribosome's moving up it, or this is moving back. And it first comes here and it says ACA. It says, hey, I got an ACA, I need somebody. Well, the tRNA shows up and says, hey, I can lock into this place. So it goes, it locks into place, and you can see on top this little ball it has right here, and it shows it right there again. It has an amino acid. So it'll lock into place, and when it does, it actually lets go of this amino acid and lets it stay inside the ribosome. Well, then what happens is another one will come, and it'll say, hey, we just got the ACA, we're done with that, but now we need a CUU. Well, matching CUU is GAA, because remember, this is tRNA. The tRNA comes in, links up, and then it connects its amino acid. It'll drop its amino acid off and connect it to this one. So now these two are connected, starting to make a chain. And so over time you can see, and see this one's going to come in and match in these. Over time you see that as each one matches in, it creates a longer and longer chain, because each time a new one comes in, it links onto the old one. And then the tRNA that lost its amino acid will go off and try and find a new one that matches it specifically. Kind of cool, huh? Well, it keeps doing this process until it gets to a spot where the protein's complete. It'll break off the chain, and this thing will form a protein and go off and get ready to be shipped off by the Golgi bodies, or the Golgi apparatus. If you remember, they package the proteins and send them where they need to go. But our proteins are complete. We did our job with the ribosomes and the DNA. And that's how you get DNA, starting here, to the mRNA, to the tRNA, creating the amino acid chain. And that's how you get a protein out of a DNA. So when you look over here, you can see really easily, we start here with the DNA, with TGT, GAA. We transfer it. Remember, we want to put A to T, but this is RNA. Remember, this is messenger RNA. So this is actually going to change into a U. So A goes to U, A goes to U. But these T's can still go to A because we can still use A here. And then we take this mRNA, transfer it to tRNA, and it's UGU, GAA, and then it'll code for a specific amino acid. We did this stuff in class of how these things pick a specific amino acid. Now here's a little trick that some kids already had picked up. When you're doing this, if you want to double check to make sure you did it right and you transferred everything over correctly, we'll simply do this. If you notice, because we switch it once and then twice, so we switch it and then we switch it back, this top strand of DNA and this strand right here of tRNA, this transfer RNA, should actually be exactly the same, except for instead of T's, you should have U's. So you notice that? TU, TU, but you have GG, GG, AA, AA. So if you're ever wondering if you did this transfer correctly, well, just look at the DNA and the transfer RNA and make sure everything matches up, except do not forget, because it's RNA, it uses U's instead of T's. So, all right, cool stuff. Well, again, if you have any questions, make sure you email me. This thing's a little bit harder, but I'm sure you guys are grasping it pretty good now. So until next time, take care. Bye.